He holds the bachelor's degree and master's degree in the life science from Handong Global University in Pohang, Korea, and earned his PhD from Department of Chemistry and Biosciences from the Karlsruhe Institute of Technology in Germany. Okay, I remember he made a wonderful talk early this year in the another conference in Seoul. So it was very enjoyable for me. Okay, so 규성조, the sharing his strategy, especially in the agricultural sector. Okay, are you ready?
Yes,
okay.
Thank you so much for your kind introductions, and also thank you for you guys joining this conference. Actually, I decided, since this conference is being held in Korea, that I'll be able to speak Korean, to share presents in Korea. Is it okay for you guys? So now, time to use your instruments. Just switch on and focus at channel 1. It should be in English from native speaker. Okay, 감사합니다. 여기 첫 부분은 좀 out of recording.
Okay, so today I'm going to be giving my presentation in Korean. My presentation is going to be about AMR, of course, and everyone here knows that AMR is very important, but what is more important is that us experts and policymakers carry out activities on this, make policies. That's also important, but the most important thing is that people are aware of this problem, not just experts and policymakers. So I am a microbiologist, and I am going to introduce what my research is about and how my research helps raise awareness to the public. The title of my presentation is Investigation in Antibiotic Resistance in Enterobacteriaceae from Fresh Produce in Agricultural Environments. The contents of my presentation is this, and as a lot of people noted, AMR is very important. In 2008, we have introduced the concept One Health. This is not just for human medicine, but One Health approach is focusing on the fact that AMR should be dealt with in many other sectors together. And here, I brought the One Health concept that is introduced in the WHO website. And in Korea, we are also discussing AMR both in humans and animals, and environment and food. In Korea, we also have a lot of projects and programs conducting research based on the One Health concept. So if we don't do anything, then by 2050, AMR could kill 10 million people per year. That was reported in the review on antimicrobial resistance commissioned by the UK government. And it's not just about the numbers. The important thing here is that we need to notice that climate change is contributing to the increase of antimicrobial drug usage, and this is leading to more death in humans. Also, during the COVID-19 period, CDC also noted that there was more than 40% of people who believed that antibiotics could be used to treat COVID-19. So a lot of people began to use antibiotics against COVID-19, and this shows that this led to at least 15% more death during the first year of the pandemic. And so I would like to talk about the global burden of AMR. In the World Economic Forum annual meeting that was in Davos this year, they had a talk about facing a world without antibiotics. So this is a very serious issue, not just in the healthcare, and you can also check this video on YouTube. It's a very important talk. It's not just about us experts in the healthcare industry, but also the economists are seeing this as an important issue. As I have told you, World Bank says that we will be losing 3.8% of global GDP due to AMR if we don't do anything by 2050. And also, in 2021, there was a U.S. report which said that 6 of the 18 most alarming antimicrobial resistant threats cost the U.S. more than $4.6 billion on an annual basis. Yes, $4.6 billion. And I am also working in Germany, but in the 16th Berlin Agricultural Ministers Conference, there was a report that was published in 2024, and they said that all sector capacities need to be enhanced to prevent AMR in both humans and animals. WHO already knows the seriousness of the issue. With the COVID-19, we have now witnessed what the pandemic can do to us. And antimicrobial resistance is called silent pandemic. So just like any pandemic, it can be a very severe issue. It's not going to be as direct and as fast as COVID-19, but it is going to come to us on a gradual pace. So in all sectors, we need to have joint efforts, and we need to have a new surveillance system or new food control programs and projects in order to overcome this issue. So this is a graph that shows where we will see a lot of death due to AMR. This is just an estimation, but you can see that Asia and Africa will have high mortality rates. Now, for the statements for One Health approach, we also work a lot with policies in our research institute. So I brought some important parts of the statements that are out there. And these are all related to agriculture because that is what I'm focusing on in our field. The G7 in 2022 welcomed the guidelines on integrated monitoring and surveillance of foodborne antimicrobial resistance. And also, in 2023, the G7 agricultural ministers gathered and announced that they support the aims of the One Health Joint Plan of Action. We have a lot of research plans, and we selected the research plan that includes the health of humans, animals, plants, and the environment as number 3 of the One Health mission. WHO and World Bank also released a report called Sustaining Action Against Antimicrobial Resistance in 2022. They are inducing a lot of people to actively participate in our efforts against AMR. And tomorrow, we were going to have the second high-level meeting at the UN General Assembly and on AMR. The title is Disruptive Innovation in AMR, a Collaborative Approach to Change. So we do acknowledge that this is an issue, but what we need to do is we need to gather energy and resources from many different sectors to resolve this issue. And that is going to be the topic that will be discussed tomorrow. So another limitation or challenge that we need to overcome is the funding commitment over the long term. And this is a report that came out in 2022, and then I will move on to some of the research details that we are conducting. So for this, we had an isolation of about 15 million samples, and it added their estimation model to analyze what is happening in the world, to see the AMR pattern that is going on in the world. If you look at the slide on the left, you can see the resistance for carbapenem. And the orange color means that already 50 to 60% of the isolates were resistant to carbapenem. And on the right, this is resistance to cephalosporin. And we didn't do anything, but already the current status showed that 70% of isolates were resistant to cephalosporin. And among the top 10 antimicrobial resistance pathogens, in 2019, we had 1.27 million global deaths, which was directly by bacterial AMR. And also, it contributed to 4.95 million deaths. Also, all 6 of the leading pathogens contributing to the burden of AMR in 2019, E. coli, S. aureus, and so on, has been identified as priority pathogens by WHO. And now I would like to show you the research trends in fresh produce and with antibiotic-resistant bacteria. So what we do is we isolate the microorganisms from the fresh produce, and then we have the sequencing and Mallet-Tauber process. And this is not just from the fresh produce itself, but we also collect samples from the soil and the water that is used to grow this. And we collect the microorganisms, and then we analyze what kind of microorganisms these are and what kind of resistance that it has, what kind of antibiotic resistance that has, and if this is also transmitted to the rest of the agricultural environment, or also if it is transmitted to the food products. So I'm also participating in this kind of research, and this is a research that was done by CDC in 2019. So in 2019, CDC, they already had this kind of picture that bacteria are everywhere. So not just about the antibiotic treatments that are being done in hospitals, but the antibiotic resistant bacteria are all over the place, included in the waters, the soils, the environments that we live in. And also looking at the agricultural front, we have a lot of the AMR that is coming from animals, and also a lot of AMR that is coming from the remnants or animal waste. A lot of the fresh produce is being consumed without being heated. So this can lead to infections or transmissions of AMR. So we focused our research on the agricultural environment, and it was based on the estimate that we would have a lot of antibiotic-resistant bacteria being transmitted in the process. So the samples were vegetables, fruits, salads, fertilizer, and water and soil. And then we had the culture-dependent method. So for these, you have to do the culturing process in order to learn if it's antibiotic resistant bacteria. And then we also use the culture-independent method. We extracted the DNA and RNA and used the metagenomic methods in order to see what kind of microorganisms were existing in the samples that we collected. And also we looked at the potentials or the possibilities of transmissions. And I focused on the conjugation test within the plasmid. If you have a resistance gene, then this can be transmitted to other areas. So we focused on the transmission. So now I would like to introduce the results that I have received. So this is not just from the agricultural market, but agricultural field, but also from the market. So here, if you look at, you can see the total number of bacteria that we have had. So we had more numbers of bacteria isolated than I have expected. We have E. coli, and which is also included in the number of Enterobacteriaceae. However, fortunately, the incidence of foodborne pathogens, such as salmonella, were not in the fresh produce was very low. And also the antimicrobial or resistant organisms were not very high or isolated in our samples. So we were wondering if it's the same case in Korea. So we worked together with the Rural Development Administration to conduct this kind of process in Korea. So our target sample was lettuce because Korean people love lettuce. We have lettuce in a lot of our foods. So we focused on the lettuce samples, the ones that are manual treated or not. So we had about 400 strains isolated, and we are carrying out further studies on this. And the fortunate result is that only 8 strains were resistant to meropenem. So it showed you that overall in the world, 15% of these strains were resistant, but in Korea we only saw 8 strains that were resistant to meropenem. So we did not have the whole sequencing yet at this level, so we just looked at the family level. So 30% of the isolates were identified as Pseudomonas. So with this, we are carrying out further studies, and I would like to show you some interesting results. So we found some novel species from the lettuce and lettuce agricultural environment. So this is included in the Scandinavium genus, and we called it Scandinavium lactucae. It was isolated from healthy lettuce in South Korea, and Scandinavium is actually multi-drug resistant. However, the Scandinavium lactucae was not highly resistant, but it also had a similarity to QNRB96 with 95% of similarity. And we also found another novel species, and we named it Liliolatu wonjuensis, and it was found in the lettuce from Wonju in South Korea. So this was very close to the type of strain that was already found in traditional Korean fermented clams. So you can see that the microorganisms that were in food could also be existing in the agricultural environment, so this can be transmitted. So now coming back to the study in Germany, with the isolates that we have obtained, we carried out further study. We selected 24, and because they did not have AMR features, because we did not have much antimicrobial resistance, what we did was we looked into the genetic features of these sequences. So we had the Crepsinolia, which was ESBL positive. The Crepsinolia has the structure genes for conjugation and also is ESBL phenotype. Also, E. coli, more interestingly, so E. coli is something that we know very well. It is a it also has the drug resistance genes. And related to conjugation, it has the conjugation structure. And the smallest had the mobile genetic genes. So with this, we did a conjugation test. And if you look at this, the plasmid, we wanted to see if it could be transmitted to other microorganisms. So we made this kind of test setting. So we used the sprouts and we made a set. And then, so at first we just used the leaves. In the second test, we made the vegetable agar, and then we used the transfusion. So we wanted to have this a very nutrient-rich agar, and we did the conjugation test. The reason that we selected E. coli is because we have a lot of recipient pathogens for E. coli. Our estimates, according to our estimates, we wanted to look at how the genes were transmitted. And if you see here, you can see that there are conjugation numbers. We had about 120 conjugations, and we had a complete sequencing, and we wanted to see what kind of plasmids were transmitted. So during the conjugation, what was interesting was we found an unexpected transconjugator. So sometimes we would have doublings, sometimes we would have smaller sizes, and sometimes we saw that smaller plasmids were created. So in the agricultural environment, especially in microorganisms in fresh produce, microorganisms could be transmitted. Then, in reality, what plasmid is existing in fresh produce? That was my question. So what we did was we made a test plate where we used sprouts. And then we use the recipient vegetables that we have used before, and then we use the plasmids that are already existing in the sprout. We found about 50 plasmids, and I want to introduce two of them. Form was about 70,000 base pair. And this has all of the features of the conjugation, and it has a multi-drug resistance. And it also has the genes, tetracycline resistance genes, as well as the additional quinoline, sulforamide, and trimethoprim resistance genes. And it also has a gene that is resistant to various metals. So, in essence, actually within the sprout there is plasmids, and the plasmids have metal-resistant genes. Then, who can be the host of these plasmids, or who could be the recipient of these plasmids? So we analyzed the metagenome. So we used the exogenous plasmid test. So we looked at what kind of hosts could be the candidates, and we found more than 50% of these called Enterochyta. And the Enterobacter cloacae, this was included as a carrier in our sample. So in the plant, we have antimicrobial resistant organisms, and this plasmid could be transmitted. That was our conclusion. So back to the Korean study, we conducted a further study on the Korean test. So we did the isolates. We used the isolates for 160 sequencing. And unfortunately, we did not find any ESBL. So in the Korean agricultural environment, especially in the agricultural environment that grows lettuce, there is no ESBL, and we had basically low antimicrobial resistance. And also sequence 11, 147, and 258, and 307, the high-risk sequence types were not detected. Then, at the Klebsiella that is found in our hospitals and in the agricultural environment, how different they are was what we wondered about. So we compare the genomics of Klebsiella pneumoniae. So we wanted to look at the antimicrobial resistance features of these two. So the Klebsiella pneumoniae isolated from hospital had very high resistance compared to the ones that were found in the agricultural environment. So you can see that these spots are concentrated on this field. So we compared the genes, and the other features of the genes were not very different, but the antimicrobial resistance feature was very different from these two. And to show you a picture of this, you can see on the right, this is the one that was isolated from the hospital. And on the left, this was isolated from the agricultural environment. So all hospital isolates possessed multi-antimicrobial resistance genes, but the isolates from lettuce and lettuce agricultural environments were not highly resistant, multi-drug resistant. So the result, the conclusion is that everyone is at risk. We all consume vegetables and fresh produce. So because the plasmids could be transmitted from what is in the vegetables, we need to control all factors and all aspects. We need to have adequate use, appropriate use of antimicrobials in the agricultural environment. We need to use an adequate dosage and also lower the use of antimicrobials.